Cher Emmanuelle, félicitations encore une fois pour le prix Nobel de chimie. Herzlichen Glückwunsch, congratulations to the Nobel Prize in Chemistry. You know we are here in the Leibniz Hall of the Berlin-Brandenburg Academy of Sciences. This has a long tradition in science, but also interaction with French scientists. The founder of the Berlin-Brandenburg Academy, Gottfried Wilhelm Leibniz, he spoke fluent French. He was for several years in France, negotiated actually with Louis XIV, le Grand Louis XIV. What does it mean to you to, as a French scientist, having won the Nobel Prize, be in Berlin and now be part of the Berlin scientific community and being a member of the Berlin-Brandenburg Academy, which we are very proud of course. You're the 80th member, 80th Nobel Prize winner of the Berlin-Brandenburg Academy.
Well, I'm very happy to be in Berlin, not that my wishes are that science goes back to the French language. I will not succeed. No, I'm very happy and proud to be in Berlin. I believe it is quite natural in nowadays that as a European scientist, one does not specifically do research in her or his home country. As a matter of fact, maybe Germany became my home country. So I'm very happy to be here as a French scientist. I feel comfortable, yeah.
You're going to stay, I hope. Or
I hope.
Are you traveling again?
No, I hope I'm going to stay. At least for me, going back to France is less natural than staying here in Berlin, because I have left France already, 24 years ago, actually.
You were born in a relatively small village, the south of Paris, Juvisy-sur-Orge, and you know, your family was not really an academic family. What made you study biochemistry, microbiology, and have the career which you actually had?
Yes, so my parents were not working in an academic field, yet they were always interested in personalities coming from academy. So my father was responsible of parks and gardens in the city, and he had studied horticulture. So I think maybe from him I got the feeling of nature and biology. And my mother was working in psychiatry, so I guess from her I got the feeling of medicine. So actually, I believe that one teacher in biology must have inspired me, because when I decided to do my master thesis in a laboratory at the Pasteur Institute, my mother reminded me that when I was about 12 years old, I came back from school telling her that one day I would be working at the Pasteur Institute. Then after, because of the French system of high school that requires to choose certain areas, already starting 15 years old. So I decided to choose biology and mathematics, also because I was qualifying for it. And then later on, during the end of my bachelor studies in biology, I decided to focus on microbiology, actually, and later on to do my master and PhD studies at the Pasteur Institute in Paris.
Pasteur is a famous institute. Actually, Pasteur is one of the national heroes of research in France, as Robert Koch is in Berlin and in Germany. And they did not get that long together, really. They were kind of, I wouldn't say antagonistic, but they were not real friends. Was this something which touches you when you came to Berlin, the city of Robert Koch?
No, I did not feel that I did not feel bad with regard to the fact to choose Berlin and continue my research in microbiology in Berlin, the city of Robert Koch. I think, yes, the relationship between Robert Koch and Louis Pasteur is just reflective of competitive research still happening in nowadays. maybe it was good in a way because it was, it's the way science goes. One needs a bit of competition, collaboration, cooperation, but in a competitive environment to push oneself to the boundaries and to the ultimate discoveries. And this is, I think, what Robert Koch and Louis Pasteur have done.
When one looks at your career, I mean, it's normal that a scientist travels around the world and sees various laboratories, but you're still very young, but you really went to many laboratories and you stayed there for a relatively long time, not just a short postdoc or something. You were in the United States, New York and two institutes, Rockefeller and Skirball Institute, and then many other places. And then you ended up in Umeå in the northern part of Sweden. You went to Austria, to Vienna, and then in Germany, actually, you went first to Braunschweig and then to Berlin. What was the reason for these many changes in university positions? Actually, you in many places you did have a more or less permanent position. You could have stayed, but you went on. What was the reason?
Yes, actually in the US I was even in 4 institutions. There was no particular reason actually, because when I started research in France, I was more formatted as a typical maybe French scientist at the time, 25, 30 years ago, with a researcher being maybe not that willing to go abroad. So the opportunity of a postdoctoral experience brought me to go to the United States. But for me, it was really a big effort to get out of the comfort zone. And once I arrived in New York, then obviously I changed completely. Mm-hmm. It was a big challenge for me. I was nearly 28 years old. I think now students and postdocs can experience mobility at an earlier age. But once in New York, actually, I just realized that, I liked very much being abroad. I enjoyed very much the international way of performing research. The reasons why I moved were mainly due to the fact, I think, that I was very fast and efficient performing the research where I was. And I had very fast developed this kind of disease that was that each time I was starting somewhere, it will take me let's say, a short time to realize what I could achieve, to achieve it, and to anticipate what I would be needed to go further. Mm-hmm. So that's why I moved a lot. I always had other opportunities, even faster than what I was predicting. And I was always evaluating the opportunities. And each time that I was realizing that an opportunity will bring me to elevate myself further and to go further in the career and to, go up the ladder faster than if I had stayed at the place where I was taking the opportunity and moving forward. Mm-hmm. So I've been indeed very productive in terms of mobility.
So you're looking for new challenges in a way. And
new challenges and to develop further my research, new ways. I have maybe this tendency to appreciate in a very short time what I can gain from a place, or I can see very fast where the place can bring me or where the place itself can develop, how the place itself can develop, and then often I realized that actually I have a better opportunity, and then I was leaving the place where I was. Yet productive, and I think every institution where I perform my research can say that they could benefit from an output from my side in terms of publications, in terms of gathering funds, in terms of mentoring a number of students and postdocs. So I think I did my job, but very, very fast at moving indeed.
You definitely did your job. Did you also, sometimes you move in science in order to learn new methodologies. Mm-hmm. Was it that way that you thought that in some places they have a methodology which you don't find in another place in order to learn new?
No, for me, it was not really the methodology that was, let's say, triggering my mobility. It was more the scientific environment, a new community. And also for me, so people are different, but for me, I have a tendency to live in the future, not in the present. At least people who know me tell me this. And I have always the wish to develop further. And when I see that I cannot develop more than, at a certain place, and I like to move around, and also it's a way to refresh myself, to reset myself, and to, in a way, it's a rebirth. So I need, on a regular basis, rebirth of myself, and moving was the way for me to give me a new challenge. But for certain scientists, they don't need the mobility for this. For me, it was just opportunities that I took and that brought me in this state of mind, I should say.
In very short terms, I mean, you got so many prizes and honors. We don't go through all of them, honorary doctorates and prizes all over the world. What was the main topic which you were interested in, and what, if you would say that in a few words? I know it's relatively complicated, and the CRISPR-Cas kind of is an enigma to many people. So what is it what you were searching for, and what did you find? What is the big discovery?
Yes, so the line of my research has always been around bacteria causing diseases in humans. And actually, I learned pretty early on that studying in this field of research would allow me to be open to a lot of areas of research. When one works on bacteria, when one studies bacteria, when one has the possibility to do really basic research on the different types of molecules and pathways which any cell has evolved to build in order to live. In addition, working on bacteria was allowing to have a little bit of insight into evolution, into the world of interactions with other types of organisms. Working on bacteria also opens doors towards the understanding of biotechnologies. If one works on bacteria involved in the environment, one has also the aspect of environmental science or plant sciences, if it's a bacterium that interacts with plants, and in my case, bacteria interacting with a human host, so really medical microbiology. And also all the applications that have evolved from research on bacteria and viruses over the years, whether this is about gene technologies such as CRISPR-Cas9, or other types of technologies that exploit bacteria for biotechnological purposes, or try to fight diseases caused by bacteria, whether it's in plants or in animals or diseases of humans. So this has always been my line of research and my interest. And I think by now we know as well that bacteria and all the microbes are extremely important for our life as a human being, none of the less because of their importance in the environment, but also because we are in a way made of microbes ourselves. And I think what is happening right now with COVID-19 reflects on the fact that microbes are of importance. They help us, but they can be also they can also cause diseases, and we understand now what we face. Working on bacteria, I've also been confronted very early on in my research on antibiotic resistance, and I think this is also one of the main challenges for the future. Here we are dealing with a pandemic with regard to a viral infection, but you have a number of infections caused by bacteria, and bacteria are multi-resistant to antibiotics, whereby it's also research in this area is essential to be able to cope with potential future pandemic that could be caused by bacteria multi-resistant to antibiotics. So this has always been the reason to work on bacteria causing infections in humans, is the line of research towards really fundamental research, understanding the mechanisms involved in bacteria and their environment, trying to understand how bacteria adapt to their environment, survive in their environment, can cause diseases if there are pathogenic bacteria, how they can evolve to acquire the potential of becoming pathogenic. So there is always this line of research aiming to develop, to understand better how bacteria function, and thereby develop new strategies for therapeutics and for treating bacterial infections. When one is working on bacteria and viruses, one is also very much interested in genetics. And the research over the last 50, 60 years have shown that actually over the past half century, fundamental research in bacteria and viruses have led to a number of findings that have been exploited into gene technologies. And gene technologies are essential because they allow to somehow rewrite the code of life. But genetic technologies are really essential to understand the functions of genes, and they are essential to understand life and mechanisms of life, whether these are molecular mechanisms or cellular mechanisms. CRISPR-Cas9 is one of these newest technology. It is transformative because it provides a tool that allows to perform and genetics in cells and organisms where the genetics was cumbersome prior to CRISPR-Cas9 technology. And this is really the genetics in higher organisms, whether one speaks about human cells or animal models or certain plants. So CRISPR-Cas9 makes a difference. What is interesting with regard to CRISPR-Cas9, as many gene technologies which have been present over the last 50, 60 years, is that it really originates from bacteria. So this is a system that exists in bacteria, also in archaea, and that allows bacteria and archaea to defend themselves against the infections of invading genomes, including viruses. So bacteria, archaea, can be infected by viruses, as we can be infected by viruses. We have an immune system, different types of immune systems, allowing us to defend ourselves against infections. The same applies for bacteria and archaea, and CRISPR-Cas is one of these immune systems. So it's a very small machinery composed of molecules of protein and RNA nature. The system has evolved into multiple subsystems, but the function remains the same, an immune system in bacteria and archaea. And the CRISPR-Cas9 system is a particular neat system, very simple, yet sophisticated enough that allows bacteria and archaea to find ways to detect the infecting virus at the DNA level and to affect the life of the virus. And this is just the exploitation of this natural mechanism into a programmable gene technology that allows to recognize a piece of DNA of interest and being able to act on the DNA and its expression that is the CRISPR-Cas9 technology. So a number of scientists, developers have now developed the technology further for multiple purposes in the domain of life sciences.
So in a way, by exploring the oldest living structures in biology, bacteria and viruses, and how they interact, you discovered a system which can cut out in a genome specific sequences and insert other sequences and use that for the most complicated human system in biology which is human and animals and plants and
exactly.
Yeah. Was this by chance that you found it, or was it when you discovered the system in viruses and bacteria? When did it occur to you that this old evolutionary mechanism is something which you can use for the most modern precision medicine you can imagine? When did you have this idea?
So actually, when one works on systems such as CRISPR-Cas that are RNA-guided systems to act on the DNA or RNA, one thinks automatically of a potential gene technology that could have potential. In our case, the fact is that in my laboratory, we were focusing on the so-called CRISPR-Cas9 system. So we were lucky enough to work on a bacterial pathogen called Streptococcus pyogenes that has this system. And my laboratory entered into the game of the CRISPR research, really focusing on this small system. So the function and the role of CRISPR-Cas as an adaptive immune system in bacteria and archaea was already hypothesized and even proven experimentally speaking. What was in need was to understand really how the mechanisms of CRISPR systems were working. And specifically with CRISPR-Cas9, we were lucky enough to have a very simple CRISPR-Cas mechanism among all the mechanisms existing. We were lucky to identify a component that is specific to this system and that allowed us to understand very fast that CRISPR-Cas9 is just a protein that is guided by a duplex of RNA molecules, and that this simplicity, if programmable, would allow scientists and developers to have a genetic tool that will be, compared to the existing genetic tool, a very simple and very easy to design, and that could provide really a large potential of, let's say, DNA manipulation if the technology was working well and would work well in diverse types of cells and organisms, which we and others have seen very, very fast after trying the technology.
So with this new technology, you can do genome editing with precision in almost every species, plants, animals, and man. You can use embryonic stem cells and modify the genome. If there's a family with a hereditary disease, you can cut out the diseased gene, insert the new gene, healthy gene, and thereby cure a disease. And tell the parents, look, this is an hereditary disease, but I can organize it away and introduce a new gene so that this will not be a diseased, sick child. This poses a lot of ethical problems. First of all, the question is, how safe is the method? And secondly, if it is safe, would you be an advocate of using this methodology to, I wouldn't say manipulate, but gene engineer, genome edit embryonic stem cells, implant them, and thereby create healthy children?
So, first of all, the technology has evolved to be specific, but the specificity of the technology depends on the genes to be targeted, and also depends on the cells in which the gene to be targeted finds itself. So there is always a new protocol according to the gene to correct, according to the cells to target, and also according to the delivery system that applies to the cell that will be corrected. So this involves, let's say, multiple checkpoints at which the precision has to be implemented. Having said this, in certain cases, CRISPR has really been developed as being very specific. With regard to the modification of the DNA of the human germline, there are still some developments to be done to really reach the precision that is necessary to have only the gene that would be to be corrected modified and not something else on the genome of the human germline.
So in the area of human application,
Exactly.
you consider this a research project?
Exactly.
But it's not something for practical use as yet.
So the technology is not ready yet to really provide this precision in human germline, first of all. Second of all, the question remains as if it is necessary to use CRISPR. I mean, nowadays, there are these prenatal diagnostics that allow to counter-select for specific mutations. And this is important to say this in correlation to the fact that CRISPR-Cas is a technology that is mainly working in our days, maybe not in research and development for animal models, but at least for human cells in being able to modify one gene at a time. So, and the number of let's say, traits are not caused by one gene, but by multiple genes. And for certain types of diseases as well, it's more complex than just modifying a gene. It depends of, the allelic nature of the gene, of the mutation. So it's more complex than one can think it is.
So you did not really overcome the complexity of hereditary diseases.
Yes.
But we have a tool to attack that and to understand it in a better way.
The technology is mainly used for really curing diseases. And I think up to now, this is with regard to medicine, the main goal of the development of the technology.
I think it's important to say, because some people think that once you have the technology, you can apply it and you can cure diseases. On the other hand, we do have in this academy, we have a working group called the Future of Medicine, Health for All. So if you think 20 or 30 years ahead of us, would you think that this would be a feasibility to think about this type of application for hereditary diseases or even cure diseases which so far cannot be cured?
Surely, I mean, there is a hope to believe that let's say, the proofs of concept that are now being in place will be able to be developed for additional genetic diseases. So right now we see patients with sickle cell disease and beta thalassemia being cured thanks to the CRISPR-Cas9 technology. So combining gene therapy with CRISPR and cell therapy. So a few patients see their lives, let's say, prolonged without the need of blood transfusion. So for the last 6 to 8 months, one has to see how the treatment evolves, and one has to wait for further clinical trials and to see how the technology can be used on a larger scale. But for sure, the technology is also developed in our days to cure certain types of cancers through immuno-oncology. It's also developed to treat certain genetic disorders such as muscular dystrophy. So it's we hope to see it develop for further serious diseases caused by genetic mutations.
There are already clinical trials with cells which have been new genes introduced,
exactly.
But this is a cellular therapy so far.
It's cellular therapies.
Okay, so
the in vivo therapies takes longer to develop, surely.
There's always a question. One is the translation of basic research to application, what can be done. But then, of course, if it's at a large scale, then scientists usually don't do that. That is the private sector who takes over, pharmaceutical companies. You have your own company, right? What do you think about scientists being involved in this type of translation from basic research to clinical application and then to private sector biotechnology development?
I think that everything became very specialized, specifically the last 10 years, and you need experts. I think that it is difficult to ask a scientist working in in the academic environment, I believe whose role is mainly to perform basic science that is so in need in nowadays to do translational research. So I think there should be scientists doing fundamental research, other types of scientists slash clinicians doing translational research, developers doing translational research, but maybe find a kind of system whereby they can work more together, or they can communicate better together, that the research performed in the basic research, that there are ways to detect earlier pseudo-discoveries that could have large applications, and that those discoveries can be taken at an early stage and propelled forward towards application. So for me, the advantage that I had is that I was exposed to the biotechnology world since already the late 90s when I was in the US. And I was always hoping that the research that I was doing could lead to an interesting discovery that could be the start of a new biotech company. And I saw CRISPR as really, the opportunity to start a biotech, knowing as well that biotech, it's also, it's also providing means for a large number of scientists, of trained PhDs and postdocs to continue research, maybe going more towards development in a private sector. I mean, there is a need for scientists to find their ways to not give up on science but continue in the biotechnology field, and also because the biotechnology environment allows to, let's say, to develop certain applications sometimes very fast, and that's why also a number of pharmaceutical industries establish partnerships with biotechnologies for the early development of the research, from the basic science to what will become a product that will be then taking forward and developed further, either by the biotechnology themselves or by the pharmaceutical industries.
If we talk about application of your technology, the CRISPR-Cas genome editing, in human, of course, the ethical problems and there are all kinds of considerations. It has to be very safe. If you go to plants, if you go to certain animals, there are less restrictions. My question would be, how much is being applied already, and how much do you see this application for the future beyond the application in man?
Yes, so CRISPR-Cas has large implications in research and development to be able to perform precise genetics. And this is really critical. I would like to go back to the type of research that we have performed to find out, to identify all the components of the CRISPR-Cas9 system and really understand how the components were interacting with one another. For this purpose, I used actually former genetic tools that I developed during my postdoctoral time, and that was allowing to perform precise genetics in bacteria. And this is important because when one uses genetics to find out the functions of genes, one wants to make sure that we look at the gene of interest and not another gene which has nothing to see with the gene we study. And for this, one needs precise genetics. So in the field of research and development, CRISPR-Cas has a large implications to really accelerate the understanding of the functions of genes, also to study better the evolution, to be able to do genetics on a broad range of variety of species. In medicine, but not only in medicine, to be able to accelerate the genetics in the large diversity of human cells to study. If you look at all the results that emerge from basic science, it's only on certain types of human cells. in plants, it has large implications as well with regard to the development of the technology to produce new types of crops, which I think are important to consider with regard to the challenge of the climate change, and to be able to anticipate the production of plants that will sustain the challenge of climate differences. And so it has really large applications in biotechnology, research and development in general.
Merci beaucoup, Emmanuelle. When Alfred Nobel spent all his fortune on the Prix Nobel, the Nobel Prize, he said he wants to give the money to somebody who discovered something which the world and the people can benefit from. And I think your research really is something where people can benefit from immediately. We already talked about clinical trials, so there's some application already in man, and in plants and in animals, of course, it's already almost in daily use, isn't it?
Yes,
so,
indeed.
Thank you very much, and this was wonderful and great to have you here in Berlin. And
thank you.
Thank you.
Thank you.